Hello everyone. Uh, in this lesson, I will talk about the structure of DNA. Okay, it's section 8.2. DNA, we know that it's an abbreviation for deoxyribonucleic acid, okay, which is the genetic material. It stores all of the informations of living organisms. Okay, if we will come to the structure of DNA, we know that DNA is a polymer. Okay, polymer. It means it's made up of very small units, which we call them monomers. Okay, and the monomers of DNA, all of us we know that, which we studied before in chapter 2 in the macromolecules. We know that DNA is made up of nucleotides. So, how many nucleotides do we have as a number in DNA? We have billions of nucleotides in a DNA molecule, just in one cell that contains a DNA molecule. It's made up of billions of those nucleotides. Okay, but how many types do we have? Okay, as a type, how many types of nucleotides do we have? We have only 4 types of those nucleotides, and we know that these 4 types are adenine, thymine, guanine, and cytosine. Okay, so DNA is made up of nucleotides. And what we have to know about the nucleotides, we have to know that each nucleotide, okay, I'll write it by N, it means nucleotide is made up of 3 parts, okay, of 3 parts, which they are, and the first part is a phosphate group, and the second part, as we understand from its name, deoxyribose, it's a type of sugar. Okay, so the second part is a ring-shaped sugar, which we called it deoxyribose. Okay, this is the sugar that's found in each of the nucleotides. And the last and the final part that we have is a nitrogen containing base. Okay, nitrogen containing base. Okay, it may be adenine or thymine or guanine, or maybe it is cytosine. Okay, these are the 4 types of nucleotides that are found in DNA. Okay, but we have billions of these nucleotides. As a number, we have billions of these nucleotides. So guys, as now we know about all of these four nucleotides that's found in the DNA, but at the beginning, scientists are confused about the structure of DNA, how it can be the same four nucleotides can be found among all the organisms. So, by 1950, a scientist named Chargaff. Okay, let me write for you. Chargaff. He he did he did some experiment, and he changed that thought among the scientists, okay, about the structure of DNA, about the four bases, and he found that the same four bases, these four bases, adenine, thymine, guanine, and cytosine, are found in the DNA of all organisms, but the proportion of these bases are differ from one organism to the another organism, okay, and he found that always adenine makes pair with thymine, and guanine always makes pair with cytosine. Okay, as you see, I am drawing two bonds between adenine and thymine, but we have three bonds between guanine and cytosine, and this is, we called it Chargaff's rule. Okay, Chargaff's rule states that what you have to know. He said that the four bases that found in all the same four bases are found in the DNA of all organisms, but the proportion of four bases differs somewhat from organism to organism. Okay, and in the DNA of each organism, the amount of DNA approximately equals to the amount of thymine, and the amount of guanine equals to the amount of cytosine. So now let me give you an example, okay, how we can find the approximately the amount of those nucleotides in a DNA sample of any organism. So let me ask you, for example, if we have a DNA sample, okay, and if you know that the amount of adenine, okay, is 31.5, okay, this is for adenine. So the question asks is what's the amount of thymine, guanine, and cytosine? How you can find it? Okay, so what I prefer, please stop the video and try to solve it by yourself. If you could find it, then it's very good. If you couldn't, then you can continue to watch the video, and you can learn how we can find the amount of thymine, guanine, and cytosine if we know just the amount of adenine. So we said that adenine always pairs with thymine. So if the amount of adenine is 31.5, so approximately the amount of thymine is 31.5. Okay, this is just for adenine and thymine. 31.5 plus 31.5, it will be 63. Okay, so how much remained? As we see, we will do this, 100 minus 63, it will be 37. But 37 for what? For both guanine and cytosine, for both of them. Okay, so we will divide 37 by 2 and it will be 18.5 for guanine and 18.5 for cytosine. Okay, by that way we can find the amount of each of the nucleotides or DNA bases in a DNA sample. And guys, regarding the form or the figure or the structure of the DNA, many scientists studied the structure or the form of DNA, like Watson and Crick, Rosalind Franklin, okay, all together or separately they worked and they found that the DNA has a double helix structure, okay, double helix structure. Okay, it means it's made up of two strands. Okay, this is very important for us because we need all this information in the process of DNA replication. As you see, DNA is made up of two strands, or it's a double helix in which two strands of DNA wind around each other, like a twisted ladder, we can say. Okay, and the strands, guys, are complementary to each other, that they fit together and they are opposite of each other. How? For example, if we have adenine, thymine, guanine, cytosine. Okay, if you have this sequence of DNA. On the other strand, we will have adenine. We know that's pairing with thymine. Thymine pairs with adenine, guanine with cytosine, and cytosine with guanine. So as we see, these two strands are opposite and they are complementary to each other. Okay, and this is what we call double helix. Okay, so guys, the last point that we have is base pairing rule. Okay, base pairing rule. Okay, it's a rule in which it states that adenine always pairing with thymine and guanine is pairing with cytosine. And what we have to know in basic base pairing rule, it says that the bond that's present, please listen carefully, the bond that's present between the nucleotide of the same strand, okay, the nucleotide that I wrote in green color, all of them, okay, the bond that we have here is covalent bond, and the bond that we have between the nucleotides of the same strand, of single strand, is covalent bond. So there are like bonds between the nucleotides, and the bond that we have between the nucleotides of different strands, okay, between the two strands, this adenine with this thymine, okay, and this thymine with this adenine, this thymine with this adenine, guanine with this cytosine, which they are on two different strands, okay, and cytosine with this guanine. These bonds that I draw in red color, they are hydrogen bonds, okay. And what we have to know, the last point, guys, we have two hydrogen bonds between adenine and thymine, while we have three hydrogen bonds between guanine and cytosine. Okay, these are very important that you have to know about the structure of DNA. Thank you so much for your listening. I hope all you understood